Perfect.
El IICA le da la bienvenida a este evento y agradece su participación. Estas son algunas recomendaciones para mejorar la calidad de la videoconferencia. Habilite su micrófono solo en el momento que le den la palabra. Es importante mantenerlo en silencio cuando otra persona está hablando. Por favor, tener encendida la cámara en todo momento para que otras personas lo puedan ver. Si necesita atender algo personal, puede apagarla y encenderla al volver. Agradecemos por favor poner en silencio sus teléfonos celulares para evitar alguna interrupción en la videoconferencia. Recomendamos cuide su postura y su posición frente al computador. Para esta reunión contaremos con interpretación simultánea. Es muy importante que cuando ingrese al enlace de Zoom del idioma de su preferencia, debe hablar en ese idioma. Por ejemplo, si ingreso al enlace de español, debe hablar en español en todo momento. Al finalizar el video, les pedimos el favor de hacer silencio durante 30 segundos. Una vez finalizado ese tiempo, el moderador o conductor puede iniciar la reunión. Muchas gracias por su atención.
Okay, well, on behalf of my colleagues, Sergio Paulo from Canada and Jose Lopez Arroyo from Mexico, I would like to welcome everyone to the Plant Health Task Force's now third annual virtual webinar series. This is a two-day event webinar series, and today we're going to be focused on mitigating the effects brought on by climate change on wheat. And on Thursday, at the same time, will be on grapes. So we hope you can join us also on Thursday. So now it is my great pleasure to introduce Dr. Stephen Kappas, the current president of the Prosa Norte Board of Directors, to give some opening remarks. Dr. Kappas?
Thank you, Dr. Winmer. And it's also my pleasure to invite you to the Prosa Norte Plant Health Task Force work. As Tim had indicated, we've had a couple of these previously, and they've been very successful. And I want to thank all of you for joining us on this particular one. So with this particular webinar, we want to highlight the mission of Plant Health Task Force. And it's to share knowledge on how to sustainably manage insects, diseases, nematodes, and weeds that negatively affect crop production in North America. And one of the most important crops in North America is wheat. So the three countries represented on this call, US, Canada, and Mexico, we produce over 80 million tons of wheat in 2020 and 2021. And we know that this is not only important for North America, but for the world, since these three countries export over 50 million tons of wheat. And we know one of the challenges to continue producing that wheat is climate change and its impact on plant health for wheat production. And that is caused by excessive heat, drought, and flooding. So today you'll hear speakers from these three countries give presentations on their research activities related to mitigating the effects of climate change on plant health of wheat. And I'm very happy to turn it back over to Dr. Winmer. Thank you.
Thank you very much, Dr. Kappas, for those opening remarks. And just before we begin, I would really like to thank Daniela St. Victor and Gloria Ramirez from MAICA, really for all of their help in putting this all together. And so now for our first set of presentations, I would like to turn it over to Sergio Paulo for his introductions. Thank you, Sergio.
Thank you, Tim. And I'm glad to be here, and I'm glad to have the honor to introduce the first speaker today. So my name is Sergio Paulo. I'm a director with Agriculture and Agri-Food Canada. and I am located in London, Ontario. I'm with the Science Technology branch. So today I'm going to be introducing to you Dr. Raju Solanayakanahalli. He comes to us from Saskatoon. He holds a lab there that focuses on understanding physiological and metabolic response of plants and their interaction with the environment which they grow. He also looks at their mechanism of tolerance to various environmental stresses, acclimation, adaptation mechanisms. And so without further ado, I will ask Dr. Raju to come on screen. And he will be talking to you about adapting wheat to arid environments.
Yeah. Hello, everyone. Can you hear me and see my screen?
We can see your screen and we can hear you.
Thank you, Sergio. Hello, everyone. I'm Raju Solanayakanahalli. I started to work on wheat beginning 2019. So whatever I'm going to present today will be relatively young research compared to some of the seniors that are present here today to speak. So first and foremost, I would like to make the land acknowledgement where I come from. The Saskatoon Research and Development Center is located on the Treaty 6 territory and the homeland of Métis. We may pay our respects to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. In doing so, we are actively participating in reconciliation as we navigate our time in learning and supporting each other. Looking at the recent climate change report that came out in 2019 with a focus on Canada, here the report highlights some of the more extreme heat, longer growing seasons, shorter snow cover periods that will be observed across the Canadian landscape. There are also projections that show that precipitation is projected to increase for most part of Canada, and bulk of that precipitation will be in the early cropping seasons, that is particularly in the spring during seeding time. However, in the months of July and August, warmer summers will increase the evaporative demands and lead to reduced summer water availability that could potentially impact some of the crops that may have lower residual moistures. Here I have some of the climate projections for Canada, particularly for the 3 wheat breeding stations. Here I have Lethbridge in Alberta, Swift Current in Saskatchewan, Brandon, Manitoba. These are the 3 key breeding stations when we look at spring wheat production in Canada. On y-axis, it goes from 1950 up to 2000, which uses some of the historical values. And these historical values were used to develop the model up to 2050 and beyond for temperature and precipitation patterns. First and foremost, when we look at the frost-free days, from the current 133 days, we will see anywhere between 18 to 20 days of increased frost-free days for all 3 breeding stations. That means we have additional 20 days of frost-free. And when I look at some of the projections here up to 2050, we see increased episodes of temperatures that hover are above 30 degrees Celsius for all 3 breeding stations. At the same time, our precipitations that are presented in the bottom panel for all 3 stations, we will see higher precipitation rate, but that precipitation decreases as the season progresses. Drought occurs quite common here on Canadian prairies. Here I have shown some of the panels for the 1961 drought, 1988, and I just skipped here the 2001 and the last year's drought, which was the largest. When we look at all these 3 panels that looked at the soil moisture index as of August 17th, that's more or less at the end of cropping seasons, we will see like 2000 drought has much larger impact on the production acreages compared to the 1988 or 1961. The 1961 drought caused the wheat production alone $670 million, whereas the 2000 drought was the cumulative effect of multiple years of having lower residual soil moisture, leading to reduced crop yields. This led to almost economic losses of up to $5 billion for the prairie provinces. In 2021, even though it was more widespread and severe, overall the yields were relatively better compared to the previous 3 stress events. That is due to results of improvement in our agriculture practices and better genetics that our breeders were able to provide to the producers. Sticking with my 2021, when we look at the number of days here on the top panel, with where temperatures hovered above 30 degrees between April and 31st, October 31st, we noticed anywhere between 20 to 30 days where temperatures were above 30 degrees. This was not unheard for some of the northern regions of Canada. And on the bottom right panel, here I'm showing the heat dome we experienced between June 29th to July 5th that coincided with the anthesis or heading period, where temperatures were anywhere between 34 to up to 40 degrees Celsius in the major wheat production belt in the prairies. And our geoclimate modeling people were able to come up with a crop stress index, which takes the ratio of actual precipitation to the potential evapotranspiration. And when we look at the major areas, here, the extreme stress was observed under 55 million hectares, and severe stress was observed anywhere between 21 to 22 million hectares. So there was significant, this led to significant yield losses. When we compare 2021 with the past 10-year average between 2010 to 2020, here we got like 48 bushels per acre, which got reduced to 34 bushels per acre. This could have been much worse if not for the better genetics and adaptation practices that farmers follow here on Western Canada. This was the premise that led us to focus on physiological traits that impart stress resilience in wheat. As I said earlier, I'm relatively new in this research. I started to work on wheat abiotic stress beginning 2019. Our lab focus, and along with our collaborators, is to look at the wheat physiology, particularly for heat and drought, and link that to water use efficiency, fertilizer use efficiency, particularly nitrogen, and linkages to the hormone physiology of it. And we still need to understand what happens to the wheat quality as a response to these stressors. And as we get more and more into the stress physiology, we are trying to utilize some of the new tools and technologies such as phenomics, spectral indices, evaluating for below ground root rates, and to have a comprehensive understanding of wheat resilience to climate change. For the remainder of my talk, I will walk you through 3 selected examples of ongoing research in our lab in collaboration with breeders and other university researchers. Here I'm showing like a set of 208 lines that were developed using Carberry and Cadillac double haploid wheat population. These 208 lines were subjected to irrigation treatment and rain-fed conditions for 6 years from 2014 to 2019. And in 2019, we were able to capture some of the physiological traits in field for canopy temperatures, gas exchange, flag leaf stomatal features, spectral indices. However, over the years, breeders had good information on grain yield and protein, and they had stored all the seeds from multi-years. We were able to utilize this grain material for further use. Here I'm showing the PCA plots where I'm showing the genotype plus genotype environment biplot interactions. Here in the inner circle of the axis, environmental axis, we see high yielding phenotypes, whereas at the far extreme left, these are low yielding wheat phenotypes. We were able to select these 6 lines along with their 2 parents, and we put them what are the grain yield under irrigated and rain-fed, the protein content, 1000 kernel weight, as well as stress tolerance index. When we look at the low yielding genotypes, the three that are listed here in white color, they had low stress tolerance index. However, the high yielding genotypes also displayed high stress tolerance index in the range of 0.7 to 0.9. Both the parents had a similar stress tolerance index. Next, we utilized some of the seed that was available from breeders and determined the kernel delta 13 carbon isotopic composition as determined by 13C and the kernel oxygen isotope composition. The combined measures of these two, carbon and oxygen in the grain, provides us a reliable information for crop on photosynthesis and transpirative performances. Here I have plotted kernel 13C and 18O against grain yield across all 6 years. The triangles represent the irrigated plots and circles represents the dryland plots, all grown at swift current station. Here, these are my high yielding lines that are very high. Higher discrimination values have higher grain yield, and lower oxygen isotopic values have also grain yield. When I combine these photosynthetic and transpirative into a ratio and look at the grain yield, our prediction capacity goes up really high, up to 9.1. And this allowed us to understand and rank the genotypes based on their water use efficiency very quickly. Then in 2019, we used this ratio and we looked at the canopy temperatures again in the field conditions, and the cooler canopies were the high yielding lines, both under stress, rain-fed and irrigated conditions. The 3D plot shows with increase in grain yield, the kernel 13:18 ratio increases, leading to cooler canopies. Particularly, I want to highlight here the line AG075, which was later commercially released as AAC Goodwin in 2020. So, utilizing the previous information that breeders had, we could say physiological breeding is definite possibility for climate resilience. And now we are funded by producer groups to look at very early generation and see whether this relationship holds good. So Jatinder Sangha, who is the main lead, we have a first year project ongoing here. Coming to the second part of my talk, where Ashwini is looking for her master's thesis, some of the heritage breadweed panel that was put together since 1842 to 2018. We have Marcus and Thatcher, which we call founder lines. Then we have beginning of Green Revolution 1960 all the way to 2018. We have lines coming from Brandon breeding program and Swift current breeding program. Ashwini looked from field-based studies, what are the wax profiles for alkenes, diketones, alcohols, and acids across all this panel. Looking at, with a focus on diketones, some of the modern lines have lower diketone levels. However, there is trade-off for having the very same lines having higher alkane contents. Further, Ashwini grew a AAC tradition, which is one of the most modern wheat variety under controlled drought, heat and heat plus drought conditions in greenhouse, and she was able to profile much more intensively here. Under drought conditions, as we see here, there was increased beta diketones, alkanes, and alcohols relative to the controls. However, heat didn't have that kind of effect. When we look at heat and drought as combined stressors, there was no additive effect. It was more of a synergistic effect resulting from heat. She also went on to look at the wax morphology, where two types of waxes were found, like the platelet types and the tubular waxes. And the platelets were predominantly primary alcohol-based waxes composition, whereas the tubules were predominantly beta diketones. And when we look at the abaxial and adaxial surface, the bottom surface and the top surfaces, here under drought, we see increased depositions of tubules, whereas under top surfaces, we see under drought and heat plus drought, more platelet depositions. Utilizing the SCM imaging, Ashwini also is currently looking at some of the trichome related aspects, where she observed on the abaxial surface with the increase in heat and heat plus drought, there was abundant trichome density, whereas under adaxial surface, it was just under drought. So she is now keen to look at what is the wax compositions of these trichomes. And we also believe that trichome may likely reduce mechanical aberration and increase the boundary layer of air to help reduce transpirations under stress. Moving to the third component, where Neha Ved, who is visiting postdoctoral fellow in my lab from University of Calgary, Neha is very much interested to look at the abscisic acid and brachynosteroid pathway and its interaction, how they antagonistically interact. Some of the recent studies in the last 8 to 10 years have predominantly focused on this pathway, and they have shown that a negative interaction between ABA and brachynosigmoid pathway do exist, such that BR pathway mutants are very hypersensitive to abscisic acid treatment. This interaction, we postulated, could balance water conservation and growth, particularly under stress. So what Neha did was she took the AC taber, subjected that to EMS mutagenesis screening, and she took 100,000 M2 population seeds and subjected them to a chemical screening where we are working with a company. They have a proprietary chemical rights on this particular chemical screening pathway that affects BR signaling. And some of these mutants show very hypersensitivity to ABA. Further, these hypersensitivity lines were screened at 10 millimolar ABA, and they were further subjected to drought screening using mannitol method in vitro, and 6 lines were selected through stringent screening. Later, Neha subjected these 6 lines to in greenhouse for under irrigation and drought treatments, where this is the AC taber, which is the parent type, and these are our 6 mutant lines. We didn't see much differences in irrigated. However, under drought conditions here, the AC taber had much lower yield compared to some of the mutant lines that were screened. In 2019 and 2020, some of these mutant lines were taken to the field, particularly into hot and dry climates that are observed in Okanagan Valley in British Columbia, where we see temperatures hovering above 30 degrees for more than 30 days. And we receive anywhere between 60 to 70 millimeters of rainfall in this area. So they were grown under rain-fed, and rain-fed was supplemented with 25% of crop water needs, 50% of crop water needs, and 100%. When we look under rain-fed condition, compared to the taber, all mutant lines showed higher yields. But in 2021, instead of using taber, we used AAC Brandon, which is the most popular and drought hardy phenotype and recent cultivar. And we subjected them again to the 3 treatments here. And our mutant lines were comparable to the modern cultivar AAC Brandon, as we didn't find much difference in yields. This is very promising for us. Coming back to the greenhouse study, here under irrigated, we didn't see any difference in the ABA content in the flag leaves. However, when they were subjected to drought, the AC taber, which is the parent line, had significantly elevated levels of ABA compared to all the mutant lines. So this indicates that either the activation of ABA independent pathway that may be related to the brachynosteroids, or there may be an activation of downstream pathway here. Since we haven't analyzed the whole brachynosteroids yet, so we are just like also giving this hypothesis that there could be a role for downstream of ABA pathways. At the same time, Neha is also looking at some of the ABA and BR pathway related genes under well-watered and drought conditions. Here, I'm just showing the transcriptome data for line 17 that has high potentials. And this understanding these gene networks allows us to better decipher the ABA and brachynosteroid pathway crosstalks and further screen for drought tolerance. With that, I would like to thank our funding agencies who have supported our research in the last 3 years. I thank my colleagues at Agriculture Canada, particularly the breeders, physiologists, modelers, as well as technology transfer people that are part of our proposals, and my collaborators at University of Calgary, Toronto, and Saskatchewan. With that, thank you very much for this opportunity.
Great. Thank you so much, Raju. That was great. A lot of information on that presentation too. I'm
Thank you,
glad to see
Sergio.
some of the great work that some of the postdocs are doing and the students. So with that, I guess we can open it up for some questions. And we have been requesting that questions can also be added via the chat.
Okay.
I see we have a hand up from Camille.
So Raju, your mutants, do you have any knowledge as to whether or not your ABA hypersensitive mutants were acting via reduced water loss through transpiration, or whether they're affecting, adaptation, some other adaptation to drought stress?
Here, based on the chemical screening we are using, we are mainly targeting for the ABA independent pathway, which will be driven through Brachynosteroids. Since we haven't analyzed that Brachynosteroid compounds yet, so that's why I'm inferring like it could be downstream of ABA until I get that results. So I would go with like it could be. So let me put it that way. Thank you, Camille.
Thank you, Reggie. Thank you, Camille. So we also have a question on the chat. I'll read it out loud. It's from Tim, and it's asked, did you look at how might these be impacting stomatal openings?
Yes, good question, Tim. Yes, we have. I haven't, in the interest of time, I didn't show some of the gas exchange measurements we have taken in multi-year greenhouse studies, where we looked some of these mutants also like under irrigated conditions. They show very good high levels of stomatal conductance, but when it comes to the drought conditions, so some of them still retain like higher levels of stomatal conductance. So we are like looking, they could have a better root system or better way of like maintaining like the water potential in the leaves.
Perfect, thank you. And I see Tim has expressed his thanks via the chat for your response. Thank you, Reggie.
Thank you.
Do we have any other remaining questions?
I see two participants raise their hand here, but I can't see them. So
let me see. It's not showing up on my screen.
It showed on my screen. If they are here, they can speak up or put their question in the chat.
And just as a reminder too, if you have additional questions, you could always put them in the chat. I just ask, please include Raju and then the question as we move on to the next presenters later, in case you have questions later on.
Keshav?
Hello, Raju, can you hear me?
Yes, I can hear you.
Yeah, I do not have this chat. My chat is disabled somehow. So it's very nice work, Raju. Thank you for presenting your work. I have a question about stress tolerance index. So I'm just wondering how you have calculated and compared that stress tolerance index.
Well, we utilized like for calculating the stress tolerance index, what was the yield under irrigated condition minus droughted divided by the droughted one. So that equation gave us like the ranking of genotypes. What was the yield reductions as a response to stress here? Okay, so I should be able to share you the formula and some references.
No, definitely. Thank you. I was thinking it's maybe coming from reflectance, but it's not that, right?
Yeah.
Okay. Thank you.
Do we have any remaining questions? Going once, twice, 3 times. Raju, I think you're off the hook. Thank you so much. That was a great presentation. Really appreciate it. We are doing really well for time as well. So if I think we can move into the next presentation. I'll just double check if Dr. Sylvie, if you're online with us, let us know if you are ready.
Yeah, for sure.
Okay, I'll give a quick introduction then. Thanks again, Raju. So let me start by introducing Dr. Sylvie Cloutier. So Dr. Sylvie, her research focuses on genetics, genomics, and epigenetics of wheat and flax. She currently leads a pre-breeding program in wheat to identify new sources of disease resistance for using basically from wild relatives, using genome-wide markers and genomic selection approaches. She coordinates a national phenotyping program in both winter and spring Trichome Morphology and A. aegilops species. And she currently co-leads the Genome Canada 4D wheat project. So Dr. Sylvie, welcome. I believe you are going to be presenting to us a presentation on tapping into wild relatives to improve wheat. So looking forward to it.
Okay, I think we're on. Let's see. All right, so good afternoon, everybody. I'd like to thank the organizer for the invitation. And I'd also like, because I don't want to forget at the end, to thank all the co-authors of this presentation. This is really a team effort that I'm going to be talking about this afternoon. And it would not be possible to run a pre-breeding program without the input of all of these people. So the title of my talk this afternoon is called Tapping into Wild Relatives to Improve Wheat. So why wheat? I think everybody here knows that wheat is one of the most important crop of the world. It contributes 20% of the calories and the proteins for the human population. And you probably also know that the world population is expected to reach somewhere around 9 billion by 2050. And with this increase in population, this means the bottom line is that we require about 50% more wheat than we currently produce. The good news is here, and it is that the yields of wheat have steadily and almost linearly increased over the past 50 years. We don't know whether this trend will continue, but even if it does continue at this particular rate, the bad news is that that rate of gain, which is about 0.8% to 1% per year, will be insufficient to feed the population by 2050. To this particular conundrum, we now have to add the climate change, unpredictability of the impact of the climate change on both biotic and abiotic stress. And I think we'll hear a bit more about that later this afternoon with some of the talk, particularly on the biotic stress, I noticed. So the end result of all of this, the elements on this slide, is that we are really looking at this gap in food insecurity. I don't like to call it security. I think it's called insecurity, which is translate into food shortage, and food shortage translates into price increase. So the conclusion of all of that really is that perhaps it makes sense to explore a new avenue of research, because the status quo ain't gonna cut it at the end of the day. And this is where I propose to explain to you how I believe that exploitation of the vastly untapped genetic reservoir of crop wild relatives might be one of the solutions. So before we really get started any further, I wanted to describe three particular terms so that we're all on the same page. So the first one is breeding. So what is breeding? Breeding is the science-driven creative process of developing new plant varieties. So here we're talking about crops. In pre-breeding, that is using crop wild relatives in crop improvement. Okay, so the end goal is still crop improvement, but the germplasm is quite different. And crop wild relatives are defined as wild plant species that are genetically related to cultivated crops. So now that we know that, this is a snapshot of the evolution of wheat. I want to keep things simple and not go into too much detail here. So let's just say that there are two main types of wheat. On the left-hand side, you have breadweed, and on the right-hand side, you have pasta wheat. And they evolved through hybridizations over time. Just don't know if you can see my mouse here, but of a cross about 500,000 years ago between red wild einkorn and goat grass, which produce wild emmer wheat, which is a tetraploid with two genomes, the A genome and the B genomes. Each of these genomes has 7 pairs of chromosomes, so this is a 14 pairs of chromosome type of wheat. It got domesticated into emmer wheat, which very recently, 10,000 years ago only, crossed to Aegilops tauschii, which is Tausch's goat grass, to produce bread wheat, and pasta wheat was derived from the AABB. So the end result is that your bread wheat on the left-hand side is a hexaploid with the A, B, and B genome, and this will become important as the talk progresses to understand that, each with 7 pairs of chromosomes, so 21 pairs of chromosomes, while the pasta wheat on the other side has the A and B genome and 14 pairs of chromosomes. So why are we looking at crop wild relatives to improve wheat, since I showed in the first slide that we've made some significant progress? Well, again, that's because the genetic diversity in elite cultivars, which are located on this side here, you can see it is much smaller than the genetic diversity of wild relatives on the other side. And this is because there has been bottlenecks over time, okay? And when I say time here, I'm talking about evolutionary time scale, okay? And these bottlenecks can be simplified into 3: the polyploidization or speciation bottleneck, the domestication bottleneck, and the breeding bottleneck. So this, the goal of pre-breeding is to therefore recover some of the genetic diversity that was lost and that remains into the wild relatives, and to reintroduce it into the elite germplasm. So this leads us to 2 specific questions that I would like to answer in this talk. And the first one is, does the genetic variability that has been lost have potential for wheat improvement? And if we can answer yes to this question, then the second question is, how can we identify it? And then how can we transfer it to wheat effectively? And so let's answer the first question here. So this is a table that I've adapted from a publication, a recent publications, and it basically shows, I chose this particular example because this is the plant health task force. So this one really shows different pests of wheat and species of Aegilops. Okay, so this is a particular genus that is closely related. It's considered a crop wild relatives of wheat, and all the genes that have been identified for these various pests. So it goes, as wide as, disease such as fungal disease like leaf rust or stripe rust, to cyst nematode resistant genes, eye spot or Hessian fly. All of these genes have been discovered in looking strictly at the Aegilops species, for example. So I think it is clear that there is a great deal of potential for wheat improvement in crop wild relatives. So in practical terms, how do we access this genetic diversity? And the rest of my talk will be aiming at answering the second question, and do we transfer it to wheat? So this is an area that falls within the scope of what is called pre-breeding. And there are actually 3 distinct phases to successfully implementing a pre-breeding program. So the first one is a discovery phase that involve activities such as genotyping, phenotyping, and data analysis. So phenotyping is really the traits. Genotyping is markers, DNA polymorphisms. And then the transfer phase, also called the translation. So how do we move, from this discovery into translating it into something that can be moved to wheat? This is usually done by crossing, but there's also potential for transformation and genome editing there that I will be discussing a bit later. And then there is the deployment or the adoption phase. And that again, that last phase falls really within the scope of breeding programs. That usually involve additional crossing by the breeders and usually large-scale phenotyping. So in the past, pre-breeding was what I would call a lonely venture. So usually you would have one pre-breeder and one breeder, and the pre-breeder would usually do the discovery strictly based on phenotyping. So they're looking for, my favorite example, the frost resistance. So they would do it phenotypically, identify that strictly that way. Today we have modern pre-breeding, and this modern pre-breeding looks quite different from the type of pre-breeding that we had in the past. So first and foremost, it is pan-Canadian, effort, and it includes people with many different specialties. So we have people who are molecular geneticists, pathologists, chemists, bioinformaticists, and breeders that are part of the team. It includes, the program includes various activities, so not just phenotyping, but it also includes genotyping and bioinformatics. Relies heavily on these activities. It is national in scope, as I mentioned. And the goal is to distribute the germplasm to many breeding programs, not just a single outlet. Therefore, this organization, structure of this pre-breeding program aims to achieve more efficient and precise gene discovery and transfer, and also to ensure a greater uptake of the discovery by the breeding community. So let's start with the discovery phase. So the first phase of the pre-breeding. And here I'm going to give an example of leaf rust, which is one of my favorite traits, where we have used GWAS, which stands for Genome-Wide Association Study. So this research was conducted by one of my master's students, Fiza Fatima, and it was a close collaboration with Dr. Brent McCallum, who's a leaf rust pathologist at the Morden Research Center of Agriculture Canada. So as a disease, leaf rust can cause 10 to 15% loss, both in yield and quality, and it's a disease that affects the wheat worldwide. So here what Fiza did, just to give a quick overview of what GWAS is. So she first started with a germplasm, and this germplasm had the 210, two different panels, a spring panel and a winter panel. In the spring panel, she had 210 different accessions, and in the winter panel, we had 160 accessions. So there's two components to GWAS, because the end goal is to do a marker trait association. So for the marker side, she had 20,000 or so single nucleotide polymorphisms that were derived from running all of this material onto the 90K Infinium array. And on the phenotypic side, so on the traits, we had two types of traits that we looked at. So at the bottom there is your typical, we looked at the field severity of the two panels. So they were grown in the field at multiple locations over multiple years, and evaluated for the degree of severity for the disease incidence. And then the other thing is Brent McCallum did inoculation with single races. So we used 6 different races of Puccinia triticina, which is the fungal pathogens that causes leaf rust, and evaluated the reaction types for each of these races onto the entire collections. So before I move on to the results, I wanted to show this because this is a phylogenetic tree that she constructed using the 20,000 single nucleotide polymorphism, the 20,000 SNPs from the data set. And it clearly shows in the green circles here, whether you're looking at one type of phylogenetic tree or the other one, that the Agelop species are related to wheat, but they're not really exactly all very close relatives. So there is quite a distinction in there between these Agelop species and the Trichome species that are in blue and teal colors. So now to the results. So she identified a lot of marker trait association with her study, but here I'm going to show you what we consider to be the major one. So if you look at the third column, you see the R square. So R square is what proportion of the genetic variability can be explained by certain markers or certain position of the genome, certain genes, whichever way you want to think about it. So for infection types, so this is when we inoculated with different types of leaf rust races, she identified 22 different loci on the wheat genome that were considered major. So that means that they explain anywhere from 5 and 35% of the genetic variability for the trait. For the spring panel, she identified 33, and for the winter panel, she identified 8 of them. And so then she positioned all of the markers that she identified with the multiple models that she used. So all together, she used 7 different GWAS models. And so this is illustrated here. So this is the 21 chromosomes of wheat. And you can see where the known leaf rust genes are positioned. So they are labeled on there. And the other thing that you can see, again, I'm not quite sure whether you see my mouse here, is that there are stacks of little dots at these various positions on these chromosomes. So if you look at chromosome 5A, for example, at the very end, you will see 2 stacks of little orange dots. And what this indicates is that we identify 2 loci there, but there were never any other leaf rust genes identified in this location. So these are new genes for leaf rust. And then if you look just below on chromosome 5B, you can see that these genes here and the markers, so the yellow dots correspond to the same position as LR18. And that means that we were able to pick up genes that are already known and identify them with this particular approach. Now, if you look at the very top, at the third one from the top on chromosome 1D, you're going to see a stack of blue dots. And what this identifies are genes that are strictly identified or came from the Agelop species. So those are resistant genes that are not present in wheat, but that they are, it's hard to explain. So the resistant version or alleles is not present in wheat, but there is a version of it that is susceptible that is present in wheat. So it is possible to transfer it to wheat using various methods, and I will get to the transfer later. So I think all in all, this is basically a summary of FISA's research here, and you can see that there are a fair amount of novel genes that we can discover from our research, both from the Trichome and from the Agelop's gene pool. I hope the last example illustrated something clearly, and that is that gene discovery relies heavily still on reliable phenotyping. And so in order to get reliable phenotyping, we need to be performing this in the fields and indoor, if possible, over multiple years and in multiple environments. And this has been very, very key to be able to achieve this research. And so under the aegid of both the Canadian Agricultural Partnership and more recently under the 4D Wheat Project, we are able to evaluate the pre-breeding germplasm, and we've done this at 5 different locations in Canada, all the way from Charlottetown in the eastern provinces to Lethbridge, Alberta, for 4 diseases and several agronomic traits over multiple years. And this long-term commitment is really, really important to this type of research. So now I'm going to move on to the second stage, which is the transfer stage of the pre-breeding. Oh, there we go. All right. Some people will argue that pre-breeding is restricted to the phase that I just talked to you about, i.e. the discovery phase, and that transfer is a completely different can of worm, if you wish. But at the end of the day, if I restrict my work to the discovery phase and there is nobody to pick up with the transfer phase, then there will not be any impact to the discovery that are made. So I've taken it upon myself to make sure to fill in that what I call the bridging team in the middle there, or the translation phase as well, because if you talk to breeder, and you will know, what they want to look at is what you can see on the right-hand side figures there. And what I'm looking at on most days is what you can see on the left-hand side, the figure on the bottom there. And therefore, if I am to provide breeders with, germplasm that look like what's on the left, they are quickly going to stop being interested in looking in pre-breeding material. So it is very important to be able to transfer these resistant genes, for example, into what I would consider to be semi-adapted material. Okay, stuff that starts to look like wheat, so that breeder can directly start incorporating them in their program. They're not against making crosses, but they don't want to make crosses and back crosses that are highly challenging. And so now I'm going to explain to you how we do transfer some of these genes that we've discovered. And I'm going to start with, and I will put this in quotation mark, the simple case of using synthetic hexaploid wheat. So this is a method that is possible to use if and only if you identify a gene in Aegilops tauschii. If you remember one of my first slides on the evolution of wheat, you remember that the last cross that took place to produce breadweed was the cross with the D genome, so that's Aegilops tauschii. So by and large, all we are doing here is that we are reproducing or resynthesizing wheat as it occurred in nature 10,000 years ago by crossing an AB, like a tetraploid wheat, with an Aegilops tauschii. So this produces an ABD, type of genome. This needs to undergo embryo rescue and chromosome doubling, which creates what is called a primary synthetics. So this plant actually has now all the chromosomes of the tauschii, all the chromosomes of the AB, the tetraploid wheat, and is completely fertile and can be crossed to Trichome aestivum, which is wheat. And then you can continue crossing and back crossing for a few generations to elite variety in order to recover what is called synthetic derivatives that now starts to look like wheat. And at this point in time, some of the lines will carry the gene of interest, other may not. You can just rescreen for the genes that you wanted to transfer, and the breeders are quite capable of taking this material on and moving forward with that. But how can we transfer genetic diversity from crop wild relatives to wheat efficiently if this does not come from Aegilops tauschii? So this slide here illustrates the complexity of the genus Aegilops. Okay, so you can see that there are some plants that are diploid, some that are tetraploid, and some that are hexaploid in Aegilops. But the one thing I want to draw your attention to is that none of these genomes except the D genome is common with breadweed. And so therefore, that means that it's a little bit more complicated or challenging to transfer any resistant genes or any genes whatsoever identified in most of this germplasm into wheat. And so this is illustrated here by the concept of gene pools, where you have primary, secondary, and tertiary gene pools, and depending on the species and their relatedness to the genomes of wheat, the A, B, and D genome, they belong to these three gene pools. And of course, the further away, i.e. the less in commons you have with breadweed, then the harder it is to cross. So this brings me to my favorite, or if you want my least favorite topic, is that crossing is the bane of pre-breeding. So in fact, transfer from the secondary and the tertiary gene pool is absolutely riddled with challenges, okay? So this germplasm has very small anthers, for example. Sometimes it has very little pollen. It is very hard to time your crossing to make sure that your male and your female parents are ready at the same time. There's very low crossability between them, particularly if you're dealing with material from the tertiary gene pool. You still have to use embryo rescue and chromosome doubling in order to get something viable. And even if all of this, you end up being successful in your crossing, you will still end up with issues such as aneuploidy, which means that you don't have the right chromosome number. You're going to end up with sterility. And linkage drag will always plague your germplasm. So at the end of the day, back crossing becomes essential to, one, restore fertility, break linkages, and restore adaptation. There are a few tricks we can use. We can use materials such as crocus germplasm, which is a high crossability wheat line, or you can use something like Chinese spring PH1B mutant, which allows you to basically break linkage drag to some extent. Nevertheless, this is an absolutely multi-year process, and there is never any guarantee that you're going to succeed. So what are the alternatives to doing these complex and difficult and challenging crossing? This is where I think there is a lot of room for gene transfer technology to improve the portion of the transfer, particularly for species of the secondary and the tertiary gene pool. So what I'm talking about here is the use of genetic transformation and of genome editing, for example. Both have the ability to bypass the lengthy and the challenging crossing approaches that I've just talked about. However, these technology also carry their own set of issues and challenges. For example, we all know that we still have regulatory issues. There's the need for gene isolation, and that is not so easy. There's improvement of these methods that still need to take place, particularly when we're talking about genome editing, for example, and there's still issues with public acceptance. On the other hand, I think it's worth continuing our effort because the advantages are great for this particular research area. You don't have crossing, no linkage drag, no sterility issues, and the genes are immediately into an adapted background. And so therefore, you don't have to do quite so many back crossing. I don't think we're there yet, but I'd like to believe that we will be able to continue researching this and provide sound science, education, and dialogue so that at some point in time, these technologies will be adopted. So these new gene transfer technologies, they all require something, and that is the identification of the causal genes. Okay, you cannot use transformation or gene editing if you do not know anything about the genes you're trying to edit or transform. So while we have made significant progress in this particular area, genes discovery, I would say, is still not a walk in the park. It's still very difficult. So before I wrap up my talk today, I wanted to touch a little bit on how we are addressing these issues now in the 4D Wheat project, which is a Genome Canada funded project that started almost 3 years ago now. So 4D stands for Diversity, Domestication, Discovery, and Delivery. And this is a glimpse of one of 5 of the activity. In fact, it's a glimpse of only a part of activity 1 of the project. But I think it's going to give you a little bit of the flavor. So here we have a 3-tier approach, where in tier 1, we are developing high quality references. So we are completely sequencing and developing high quality references so that at the end of the day, we will have all of the diploids of the Aegilops species represented at that level of sequencing. And then in tier 2, we are doing something a little bit different. One can call it reference guided, other can call it, scaffold level sequencing. And that we are dealing with all of the tetraploids and the hexaploid species of Aegilops in there. And then in the 3rd tier, we are doing sequencing on a larger number of accessions. In fact, we have sequenced 500 different accessions, but in a lower coverage. And this is allowing us to basically work with SNP discoveries on a large scale. So at the end of the day, the hope is that this is probably going to be a set of data that's going to allow us to, and I hope, to be a game changer in the pre-breeding of wheat, and certainly be a game changer in facilitating gene discoveries in the Aegilops gene pool. So on that, I would like to summarize my talk by saying that we've talked today about the world population growth and climate change challenges, the shortcomings of the current rate of gain in wheat yield, the erosion of the genetic diversity in the wheat germplasm, that pre-breeding may or may not, but I hope I've shown that it is a possible option to broaden the genetic diversity of wheat, that there are still many challenges to work with crop wild relatives that there's currently a shift in pre-breeding models as it stands from, a single person to an actual team approach. I've provided some example of current pre-breeding methods in gene discovery. I hope I have highlighted the importance of accurate phenotyping in many environments in order to get accurate data on the material we're working with, and address the potential for alternative transfer technologies such as transformation and genome editing. And at the end there, I just highlighted a few of the current and near future activities of the 4D Wheat project and how we hope that they will help us with the gene discovery portion of the pre-breeding programs. So before I finish, I'd like to thank the people in the various labs who have contributed to producing some of that data, and of course the fundings of the Canadian partners who have participated in that. And I thank you for attending this webinar. I hope that I conveyed to you that there is potential to improve wheat using crop wild relatives, and that our team approach and strategy are viable and efficient. And I would be more than happy to entertain some questions. Thank you.
Thank you, Sylvie. Dr. Clea, that was a great presentation. Fascinating research, really. Leads me to a few questions of my own, but give an opportunity to some other folks to raise their hand and ask some questions. I see one question in the chat from Tim, so I'm going to read it out loud for you. It says, are you or anyone in your group looking for drought resistance or heat tolerance genes in Aegilops to use in breeding for wheat?
I'm not, and that's particularly because I worked on drought resistance in flax, and I wish I had asked that question of the previous speaker. Are you working on roots? I worked on roots, and boy, I don't want to work on this again. So I'm not, and I know that there is interest in that, and other people are. But we had to start somewhere, and we started with diseases. And so our focus at this point in time is strictly on Fusarium head blight, leaf rust, stripe rust, and powdery mildew.
It's a great talk, Sylvie. Can I ask you one question? When you make these synthetic wheats, do you ever notice problems with increased seed dormancy? So do you find that they have more trouble germinating when you first generate them?
Yeah, that's a problem. So after you after you harvest the seed, we usually put them in the fridge for a few days, in the freezer for a few days, at room temperature for a few days. You have to be patient. I know you want to go to the next generations, but we find that a month is a good time of doing this every few days, of rotating between these three conditions to break the dormancy. Recently, we've made crosses between wild relatives and wild relatives. So Speltoides to Speltoides, and Comosa to Comosa, and so on and so forth. And I was impatient. I put them in the ground, and then it was time to put them in. We said, oh, only 6 out of 16 germinated. It was time to put them in vernalization, and we noticed that some more were now germinating. So that definitely comes from the wild relatives. There is this greater degree of dormancy there. So yeah. So yes is a short answer. Yeah.
Thank you.
Thank you. And I see that Raju also has his hand up. So Raju, go ahead.
Yeah, thank you, Sergio. As always, very great talk, Sylvie. I really enjoyed your presentation today. Coming to that heat and drought, will this material be available if we want to screen them for heat and drought tolerance here in Saskatoon?
The germplasm, okay, comes from different gen banks, okay, and come from various locations, okay.
Yeah.
So I cannot distribute it freely, but you can recover. I can distribute what it is that I have. I'm at liberty to distribute, not what I am not at liberty to distribute. However, what I'd say is that if you can get from the gen bank where the material come from, you know, because they distribute it freely under certain agreement of the treaty, then there's no problem to distribute it. The advantage of this is that we have all of the phenotyping, right? And so we can provide the phenotyping because we've developed it. Okay.
Yes.
But we have to be mindful of the intellectual properties and, you know, where it originates to and from.
Yeah, definitely. I will follow up with you on this, Sylvie. Thank you.
Perfect. Thank you. Sylvie, thank you so much. I think from looking at the schedule, and we're doing really well for time. So I think we're on track. We are on track for the next presenter. If there's one remaining question, we can take that really quick. If not, I encourage you to put your questions via chat. And Sylvie, if you don't mind, just maybe answer them in chat if any additional questions come up.
Sure.
So if there's no emerging questions that need to be raised now, I think I'm going to pass it over. So thank you again, Sylvie. Great presentation. Thank you for being here with us. And I will ask you some other questions offline later.
Perfect. Thank you. Bye, everyone.
Okay, so with that, I'm going to introduce the next moderator. So Dr. Jose Lopez Arroyo from Mexico, with also part of the Plant Health Task Force, is going to be joining us next. So Jose, are you there?
Claro, aquí estoy. Buenas tardes.
Yes, sir. Here I am. Good afternoon, everyone. I am Jose Lopez Arroyo. Proudly, I represent INSAP at the Plant Health Task Force. Today, I will be moderating the session from my colleagues from INSAP. And it is a pleasure for me to introduce as our first speaker, Dr. Juan Angel Quijano Carranza. Dr. Quijano is a graduate from the Autonomous University of Querétaro, Mexico. He is a researcher in the area of modeling at INSAP ever since 2009. He is advisor to Senacica in the phytosanitary programs. And for 38 years now, he has been working in his area of expertise, which is modeling. Today, Dr. Juan Angel Quijano will refer to changes in the risk of foliar diseases in wheat associated to climate change. Go ahead, Juan Angel. Thank you. Thank you. Thank you, Isabel. Thank you, everyone. Allow me to share my screen. I wonder you could confirm that you can see my presentation. Yes, we can see it. Okay, fine. Thank you. Well, continuing with our discussion on mitigating the effects of climate change, in this case on wheat, we will address the impacts that are already evident and those that we expect in the case of foliar diseases. Particularly, I will refer to wheat rust, which is one of the most common foliar disease. During the presentation, I will refer briefly to the connection between agriculture and climate change. We understand there is a two-way connection there, and I will try to explain that. We will also refer to the meteorological requirements of the organism that causes this disease, the wheat leaf rust, Fuchsinia siticina. What specific requirements we are aware of, or what the meteorological conditions favor of rust infection. And we will also analyze the perspectives of infection due to wheat leaf rust in Mexico, under a very likely climate change scenario, perhaps the one shared by most climate scientists. And then I will lastly share some evidence on what climate change has already caused regarding this phytosanitary problem. As to the connection between climate change and agriculture, we already know there is a two-way relationship here. Agriculture currently, especially if we refer to industrial food production systems, contributes significantly to climate change. And climate change, on the other hand, in general, we clearly understand that it causes negative effects on agriculture. What is this relationship like? Well, there is this two-way relationship because when we see food production activities, and I want to be very clear in this, particularly in industrial food production system, where the producers are just a provider of raw materials for mass consumption or for the manufacturing of other type of food stuff. Well, what we seek here is productivity. We know that the effect when the climate change effects increase, and we will represent it here in a very simplified manner, an increase in temperature and the changes in distribution of precipitation, or when the effects are worse or stronger, there's a reduction in agricultural productivity. We know there is that connection. If we increase temperature, we affect the distribution of precipitation, and that will reduce agricultural productivity. And that reduction in productivity we identify as related to water stress, to temperature stress, to nutrient deficit, and also to greater aggressiveness of pests and diseases. As a response, by fearing this downward pressure, agricultural productivity, we respond with technology to mitigate these effects. And how do we respond? Well, increasing industrial agricultural practices related to expanding agricultural land, but especially intensifying the use of external inputs. Dr. Sylvie just explained in her very interesting presentation that this struggle we have been facing for so many years through genetic breeding to obtain better quality food stuff resistant to diseases is a race where we get phenotypes with a lesser genetic diversity, but with more desirable characteristics. So the intensification of industrial agricultural practices has a negative effect on natural resources as our biodiversity, organic matter in soils, the capacity to storage moisture, humidity in soils, and increase vulnerability. Greater vulnerability of natural resources, therefore, in itself will also increase the effects of climate change. And this is, here we see this negative feedback connection between agriculture and climate change, particularly, as I mentioned, in industrial production systems. Now, referring specifically to the connection between the greater aggressiveness of organisms that cause foliar diseases in wheat and our practices, well, obviously, industrial production it is not possible, or it is harder to try to break away with this cycle using multiple varieties or low-scale production systems with associated crops. So very likely, We will continue to depend on these highly specialized systems, and therefore we would continue with this we will continue in this ongoing cycle to try to improve the resistance of our varieties, but dealing at the same time with better adapted or more aggressive varieties. So under this scenario, then, it is important to understand the organism that causes the disease. So in this case, the case of wheat leaf rust, we know it is Puccinia. And as any other organism, we know that all organisms respond to the environment. Our tool to understand this relationship is the genotype environment connection, and we know that each one of them has a range of adaptation. Each organism has a range of adaptation, and there is a range within each element that is optimal for that organism. And as the conditions change, that organism may either move to a suboptimal condition or totally move out of its range of adaptation and be left under a non-adaptation condition, or just the opposite. It would be in a suboptimal condition currently as a result of the change in conditions and move into a more optimal condition. So this is valid both for organisms that cause diseases as well as for the crop itself. In our country and in the entire world, in fact, we have seen how areas that used to be highly productive for species like wheat suddenly have come across more difficulty, drought, early frost, less favorable distribution of precipitation, showing that that area that was highly productive has now perhaps become to a suboptimal condition. So we have to address it either with planting dates or changing regions. And for the case of organisms, the same. We have the case of diseases that had disappeared for many years, and all of a sudden they start to pop up or increase in frequency. So this connection between the requirements of the organism and the changes in meteorological conditions and environmental conditions in general. Now, talking about the conditions we expect, where climate change will lead us to evolve. There is a clear example by Prank et al. In 2019, not with leaf rust, but with stem rust, where they analyze the different phases of the disease, and they analyze what could happen with each one of these phases under climate change conditions. And they say that it is very likely that there will be a tendency to increase temperature and reduce relative humidity. This could favor turbulence conditions that would allow for a more efficient distribution of spores. But on the other hand, during germination, when there is less favorable relative humidity, this will possibly affect also the rest, particularly in the rainy season, not in the case of irrigation, where water comes provided to the plant as opposed to rain-fed. And we could expect something different or something similar for leaf rust. The experimental data we have on temperature and relative humidity requirements, we see that we have small parts of this requirement. Harudi et al. In 2014 say that the optimal temperature 15 degrees C, with a relative humidity between 60 and 80%. And for this, we need at least 12 continuous hours under these conditions, as well as free water on the leaf. That is, water droplets on the leaf to favor the most optimal condition so that the rust spore can germinate on the leaf. Roefs et al. In 1994 indicated that the optimal temperature was between 16 and 20 Celsius, with a relative humidity of at least 60% and at least 3 continuous hours. And with also a free water was essential. Valaville et al. In 1995 focuses more on 15 degrees for the optimal temperature, makes no mention to relative humidity, but also says that 4 to 6 hours, continuous hours under this condition and free water are essential, indicating that high relative humidity is required. We know that high relative humidity is not a problem at nighttime. In the daytime, we could have low relative humidity, but we know that at nighttime, the crop itself generates relative humidity conditions. So we have proven this by measuring relative humidity at a weather station in an agricultural plantation and measuring the microclimate within the within the crop. And when we see that when a station in the open area marks what have you 50 or 60% relative humidity within the crop, particularly at night, we can have 20-30% more. It is important to know that. Why? Because when the wheat crop, covers the soil completely, when it is totally closed off, then overnight or at night, we will have this relative humidity condition, making the critical factor for the infection be temperature. Now, what we would like to know is the full range of response of the organism, both to temperature as well as to relative humidity. And well, what we really have is just a part of the response curves. As you saw, not all authors refer to all ranges of relative humidity, nor all ranges of temperature. Hardly anyone studies, or there are not many reports of what happens with the temperatures below 10 degrees C. So one challenge we have is to fine-tune our knowledge on these requirements, obviously using laboratory studies to complete these ranges or response ranges and have all the entire knowledge, as much knowledge as possible, to see how the organism can react under the different weather conditions that one could expect. So we said that the relative humidity is key in that night. And this is the example of a weather station in the state of Guanajuato, in the town of Abasolo, southwest of the Bajío area in Guanajuato. And we can see that during one day on March the 2nd, 2021, we can see that the relative humidity conditions occur between 4 and 7 a.m. and is marked here with this blue square, with relative humidity, which is favorable and extends, so covers about 3 hours, but temperature, excuse me, this is temperature, this is temperature, I am, no, relative humidity. Temperature here in red, we only have in the optimal range after that condition, so they do not match. The times of the day when favorable relative humidity and temperature coincide, these do not occur. There are not conditions for infection here, although each one occurs separately. They do not coincide, and therefore we could not expect the conditions for infection. But in the same location, notice this. If I move to August 26th, that same year, notice, we see that between midnight and 6 a.m., there's this complete period, more than 6 hours, where they do coincide. Relative humidity, high relative humidity, more than 60% coincides with the temperature range, the optimal temperature range between 15 and 20 degrees C. 2 minutes to finish. Okay, yes, sir. 5 minutes? 5 minutes? Did you say? Okay, fine. 5. So in Guanajuato, there is a warning system that monitors the data through the different stations, and they have been able to identify that when they verify the weather conditions, favorable weather conditions, about 5-10 days later, we can detect the infections in the field. This is being used to forecast possible rust outbreaks. So in summary of the conditions, we need at least 4 hours with temperatures between 15 and 20 degrees Celsius. Precipitation between 1 and 5 millimeters is desirable. And once the infection has started, it develops basically depending on temperature. Now, as to the perspectives for this disease under climate change conditions, we consider the RCP 8.5 scenario. For climate change scientists, they consider this is the one with the highest emissions. There's a discussion as to whether it is the most likely because it is the most severe, but many consider the most likely. And it is believed that by 2100, the CO2 concentration will reach almost 1000 ppm, determining an increase in temperature of 4.3 degrees C in average. Considering the scenario, variable meteorological data were developed daily, obtained from 2 different periods from 1950 to 2020 and from 2020 to 2100. They analyzed daily information with the favorable conditions with minimum temperatures between 15 and 20 degrees C and precipitation between 1 and 5 millimeters. The risk index for the leaf rust was created, dividing the number of favorable days over the number of days in the period. We considered 2 periods from February 1 through April 30 for the winter cycle and from July 1 through September 30 for the summer cycle. Here you can see the formula I just mentioned to generate the index. The flow chart, well, meteorological data were processed. These were input in a system that allowed to make the calculations automatically. They calculated the risk index for each year and for each node in the database. The values for the cumulative probability for each node was used, focusing mostly on the 80% cumulative probability. The data information was put into GIS database, and this was interpolated using the IDW method and developing risk maps. With the methodology, we saw that under the current conditions between 1950 and 2020, that is the current conditions, in the summer cycle, we have this condition. The most important production areas are up in the highlands in central Mexico, the states of Guanajuato, Tlaxcala, Oaxaca. And we see a prevalence here of low index values. The biggest risk is related to values closer to 1. 1 minute to end. And the lower values are at a lower risk. They would move into severe conditions. And in the case of the winter cycle, the most important areas definitely are Northwest, with Sonora, and the center of the country with Guanajuato, where we have a low risk condition. And this would show in Sinaloa an intensification of favorable conditions. Quickly, there is evidence in the variations in annual mean temperature, both in Sonora and Guanajuato. minimum temperature, however, is only evident. We only see an increase in Guanajuato, and this leads us that there has been an increase in favorable days for the infection, not so much in Sonora. And as final comments, due to climate change, the risk of wheat leaf rust infection has increased in some regions, and it is expected to increase in basically in rain-fed areas. We should not discount, or we should include what would happen with adaptation in some rust strains, particularly because it is inevitable to have more favorable conditions for rust in many regions. And it is very important to provide technical support to producers so that they can work to reduce the crop predisposition, avoid excessive irrigation, for example, high density, particularly associated to later dates, and eliminate alternate hosts, and above all, to work on fertilization rates that will not cause over-fertilization. That is all as far as I'm concerned. Thank you very much for your time.
Thank you, Juan Angel. We don't have time for questions, so we have to go ahead with our next presentation, but thank you, Juan Angel. Thank you. So following our agenda today, we now have a presentation by Dr. Elizabeth Garcia Hernandez. She is a researcher at INIFAP, and she works at Valle del Fuerte Experimental Field. She has a PhD in 2016, and she has been working with INIFAP since then in genetic improvement of wheat, focusing on rust resistance. And today, she will be talking about climate change and its effects on pathogenic variability of wheat rust in Mexico. You have the floor, Dr. Garcia.
Buenos días. Buenos días a todos.
Good afternoon, everyone. Can you see my screen? Yes, we can. Thank you. Thank you. Once again, I wish to thank the organizers of this scientific event, so important for all of us and of great international interest. Welcome to all participants. As we heard in the previous presentation by Dr. Quijano, some of the points he has covered, I will be looking at in greater detail. And if you have any comments or questions, you can do it directly at the end of my presentation or through the chat box. To begin, I have to say that wheat in Mexico is based on two main species, Trichome aestivum and Trichome durum. The first species is used mainly for bread flour, and the Trichome durum is used mainly for semolina to prepare pasta. Wheat is a cereal that is adaptable to a great variety of agroecological conditions and very adverse to optimum conditions. Most of Mexico's territory produces wheat. And the levels of technology diversify the different production areas, depending on the inputs each region has. The current situation of wheat in the world, we can say first that it is the most widely produced cereal in terms of surface worldwide. And as for production, It's second to rice only. Worldwide production in 50 years is expected to demand greater yields of this cereal because of growing population and more demand for processed foods. Therefore, increase in production should be gradual. And what we see in this chart, in this table, is that in 2020, wheat production worldwide was of 776 million tons. And if we look at this year, the forecast, instead of increasing, is set to be reduced by 6 million tons. Why is there a decrease in wheat production in the world? Well, it's because of biotic and abiotic factors. Some of the abiotic factors in Mexico include the drastic effects of high temperature on our production systems. We've had very high temperatures, but also very low ones. Then there's soils with little fertility, salty soils, excess humidity, and we've had hail and frost. These abiotic conditions cause problems in all Mexico's wheat production systems. Another important factor in production, or that has led to these decreases in production, are pests and diseases, the biotic factors that can have a variable effect depending on the area. Generally, we see that losses in production because of insect pests in Mexico are at around 15%, mainly different species of insects that affect wheat, but then we also have weeds that lead to 10% losses in certain areas. Of course, we might have more weeds than pests in certain areas, so these are all average figures. And I will focus mainly on diseases, the effect of diseases, and mainly rusts that affect wheat. And we see that they affect 30 to 100% of our production systems. The most common diseases we see in terms of rust are yellow rust, stem rust, and leaf rust. These are three pathogens that have lived with this crop throughout the years and historically have caused global epidemics that have been very serious. Wheat rusts are caused by fungi of the Puccinia genus, which have historically caused important economic losses in some of the varieties of wheat and have even led to worldwide emergencies. These fungi can reproduce quickly and in large scale, and they can also create great variability throughout cycles because they mutate and have genetic recombinations. That means there are new physiological races of the organism, and it makes varieties susceptible. Yellow rust in the latest years has been the most predominant disease in wheat crops in Mexico. Our national improvement programs have focused on constant monitoring for this pathogen and the varieties in different production systems. The yellow rust shows small yellow lines on the leaves, and that's why it's called yellow rust. Here are the races present in Mexico until 2002, mainly three described races. The optimum temperatures that served them, or the cycle of this pathogen, were between 5 and 15 degrees centigrade. And these are actually low to cool temperatures. Therefore, yellow rust was mainly found in areas that had these temperatures. This table here shows that yellow rust during the 70s, according to research and the literature, it required 12 to 20 degrees centigrade to complete its cycle. However, we know that some of these pathogens and races have adapted to warmer conditions and drier or less humidity conditions. In 2013, we had a national study with samples taken in the main wheat production areas in Mexico. Samples were collected and processed in our National Rust Laboratory, and we found 13 physiological races of yellow rust. These races were distributed throughout all production areas in both warm and cooler conditions. This means that rust, and here we see the pictures of the different locations in Mexico where we did not have rust until before 2001. And after these studies, we found different races in areas that did not have it before. And here we can see that yellow rust has adapted to warmer climate conditions. It is no longer limited to colder temperatures. Here we see a picture of yellow rust with strong incidence in our northwestern region. In the conditions we have in our north, the climate tends to be very warm and low humidity, and some isolates of the yellow rust have adapted to these conditions. There are some cases even that have great incidence even from the seedling state. This is very worrying for producers and improvement programs, because if we have the variety susceptible even from this very stage, they have to find a way to manage this. Here we have a table showing the severity of yellow rust in 10 wheat varieties evaluated in 7 environments. So we see that some of these varieties, especially the first ones that were released in 87 and 96, have lost their resistance to yellow rust in almost all environments. Only the more recently released varieties like Altiplano and Nana, that used to be important varieties, had conserved their resistance up until 2010. Now, what happened in 2014? In Mexico, we found new physiological races of rust. About 80% of the germplasm that year, that was the basis for our National Wheat Improvement Program, became susceptible. And not just for flour and bread wheat, but other types of crystalline wheat as well, because it is the most used in the northwestern part of Mexico. There were also problems with one of the physiological races and the triticales as well. So that led us to pay attention to this disease. Our current improvement programs focus on the use of durable resistance so that we can protect our wheat varieties from the most races of the pathogen that we can. During 2014, and here we see a chart with the different varieties available to that date. These are for the spring, summer period. And we see that the main variety was Nana F 2007. And in 2014, we had a new race that overcame the resistance genes that the variety had. So it was a setback to our improvement program. We already had a genetic pool from that variety, and some were coming in to compete in terms of yield. So with this new race, all other new varieties had to be eliminated as they were all susceptible. So that was the landscape of the overview of yellow rust for our improvement program. Another not less important pathogen was mentioned by Dr. Juan Angel in the previous presentation, leaf rust. This pathogen has also lived throughout the years with the crop. It's considerable nationally and internationally because this pathogen is widely adapted. It is practically present in all wheat production areas in all climates and conditions in Mexico. The level of specialization of races of Fuchsinia triticina, which is this leaf rust, is different if we are looking at pasta or bread wheat. So the races that have an impact only on bread wheat do not affect crystalline wheat. So they're specific to each species. In 1994, we identified a race that has been called MCJSP in south of Sonora. In 1996, Oaxaca, another race, MBJSP, was characterized. That was the only work we had done in terms of races in Mexico, at least races that had an impact on the crop and led to economic losses. These were the only two ones that were known in the 90s. Now in 2001 in Mexico, in the north part of the country, a new race was found, BBGBN, and it had an impact on over 80% of the germplasm that was available through our Center of Improvement of Corn and Wheat. And the most popular Mexican cultivar that back then, Alter C84, showed susceptibility to the race. What happened after 2001 when we saw a virulent race of LR72? After 2001, there was a new race that also overcame resistance to another gene, LR26, and it was called BCGBN. In 2008, we had another new race that was virulent to another combination of genes, LR27 plus 31 and LR12. And starting then, the pathogen evolved more dynamically. In 2008, a new race was found that was virulent on LR3. And for 2017, one new race, BBGBP, that also affected the cultivar that was most used in crystalline wheat varieties in the north of Mexico. Currently, worldwide, we have reports of over 50 physiological races of leaf rust. In this chart, we see a study that we performed in the northern state of Sinaloa in Mexico, characterizing the isolates we collected in the field to determine the population of the pathogen that is present in this production area. This study isolated 7 physiological races present in the limited catalog of varieties that we have in this production area. And we see the frequency. CBJQQ was the most frequent at 29% of isolates that were collected. And the least frequent was MFJSP. These 7 races are newly reported in Mexico, and it's also important to pay attention to that because we want to ensure we can monitor the crops to avoid any epidemics. And now we have another important pathogen that is genetically controlled in Mexico is Puccinia graminis with tritichy in the form, which is stem rust. This pathogen is important for Mexico and the world because it is extremely virulent. It affects practically all parts of the plant, from the foliar areas to the stem, which is where it mainly sits, and it can lead to losses of 100%. In Mexico, it is currently genetically controlled since 1950 using genetic resistance. The different varieties of wheat we have in Mexico, breadweed and durum wheat, have genetic resistance. This is the most destructive disease, and the last susceptible variety we had in Mexico was Zaragoza 75. The population of stem rust in Mexico is comprised of 6 physiological races reported by Singh in 1961. And these races are found only if we grow experimental seedlings or trap greenhouses. So it's not found in any of our newer varieties. So what strategies do we have to ensure we can manage and handle rust in Mexico? It's a combined approach to ensure sustainability. We have to ensure we know the pathogen well, the hosts and environment, and when all factors combine, we will have an epidemic. And we have to establish a methodology. And that's what we're doing. We monitor the pathogen constantly. We have trap nurseries or trap greenhouses or experimental nurseries. These trap nurseries are established in collaboration with the International Center for the Improvement of Wheat and Corn. They provide some lines for us and also help with our improvement program at INIFAP. We have a nursery with 100 lines and varieties, and they are grown in the main wheat production areas. We have 3 dates: early growth, medium, and late growth, so that we can trap as much as isolates as we can. And so these trap nurseries are established in production areas. We collect the isolates when rust has conditions to provide the spurls, and we collect these. We label the variety as collected. We then take it to our national rust lab, which is in the state of Mexico. The isolates are then processed in incubation chambers, where they receive the artificial conditions necessary to finish the infection process. Then they are taken to the nursery, and there the pathogens complete their process through sporulation, and we take data in seedlings and adult plants with previously established scales, and we determine the genetic of resistance of varieties and some crossings or families in seedling state. And we also determine characteristics and traits in the adult plants. And these are all then verified with the data we see in the field to look at the severity of the disease and reactions on the field. And this all helps us determine the genes and level of resistance of each variety, and it helps us differentiate among all the different lines that we have as candidates to improve our program, which ones are truly feasible, so that we may release them in each wheat production area in Mexico. And this table now shows some of the varieties that are currently available. The Terre and Xochitl varieties at the bottom of the table are new varieties that have resistance in most of the races we've identified in Mexico. And so that's the progress we've made in terms of improvement in our national center. That's it on my part. Thank you very much. I would like to thank the Wheat Improvement Program and all the people involved. This information is part of all the work that each researcher performs on wheat in Mexico. It's not a single person. We have a whole team collaborating for the evaluation, not just of rust, but all other diseases we have in wheat, to ensure we can find healthy varieties that we can share with our producers. Thank you. Thank you very much, Elizabeth. We are finished with our time. There are some questions in the chat box. Hopefully you will be able to answer them, Elizabeth. This is the end of INIFAB presentations for today. And now we have the next part, which is by Dr. Whitman. Go ahead, Tim.
Thank you very much, Jose. And thank you for all of the previous speakers. Those are really fascinating presentations. And so we're now coming down to the last two presentations. And so it's my pleasure to first introduce Dr. Mary Gutierrez. Dr. Mary Gutierrez is a research geneticist and lead scientist in the hard winter wheat genetics research unit at the U.S. Department of Agricultural Research Service Center for Grain and Animal Health Research in Manhattan, Kansas. Her research program integrates molecular breeding technologies towards the genetic improvement of hard winter wheat for disease and insect resistance, tolerance to heat and drought stress, and nutritional and end-use quality. And so her presentation today is on USDA ARS breeding efforts to mitigate the effects of climate change on hard winter wheat in the Great Plains of the United States. So Mary, take it away, please.
Thank you, Dr. Widmer, for the introduction. Can you verify that you can hear me all right?
Yep, and your slides are good.
Excellent, thank you. Well, it's such a pleasure to follow such wonderful presentations that really have set the stage in many ways for the work that I'm going to talk about today, which highlights some of the breeding efforts to mitigate the effects of climate change on hard winter wheat production in the Great Plains of the United States. And let's see. There we go. As a brief outline, I'll talk a little bit about who we are, where we are, what we do, and share some reflections on climate change in the context of our production region. I'll talk at greater length about what we're doing to address the effects of climate change now, and what I see as some of the operational imperatives for us as an organization. So to introduce the team of the Hard Winter Wheat Genetics Research Unit, this is the lead scientific staff, Bob Bowden's a field pathologist, John Fellers works on leaf rust genomics, Weewa Bay manages our genotyping lab and works on Fusarium head blight, Ming Chen is an entomologist with particular expertise in Hessian fly, Katie Jordan is our bioinformatician, and then there's me, and my expertise is in breeding and genetics, combining field and laboratory work. And we do try and work very much as a team within our unit. To introduce to you the Great Plains and as a wheat production region, in 2022, there were roughly 9.5 million hectares of hard winter wheat planted, and only 7 million hectares of those were harvested. We have a very high rate of abandonment of our acreage. Our production was significantly down in 2022 as a reflection of the heat and drought stress that was encountered throughout the region. Among the Great Plains states, Kansas is shown here in bold, 2.8 million hectares in 2022. And the location of our research unit in Manhattan is approximately at the H in hectare. So the mission of our unit is to develop and deploy genetic solutions to economically important biotic and abiotic stress problems in hard winter wheat. It's really to find the genes and to make them available. And we typically do work to package those with marker systems to support the regional breeding efforts of the state, the university programs, and the private sector programs within the region. A little bit about climate change as we see it on the Great Plains. The images on the left focus on annual temperature compared to the 20th century average, and it's kind of a decade by decade progression from cool to really very, very hot. We're seeing really dramatic increases in average temperature over the last century. Coupled with those increases in temperature are increases in heavy precipitation events. The image on the lower right illustrates the number of severe precipitation events that have happened over the given number of years. And what we're finding is that we have more and more severe precipitation events, high rainfall events that can be very damaging to our production. At the same time, the weekly release of the US Drought Monitor that's produced by the University of Nebraska at Lincoln is like a prominent feature on my Twitter feed. The region has really been characterized by drought. So it's kind of this paradox that we have these really severe high rainfall events that impact our production adversely, and at the same time, we're encountering very profound effects of drought. Southwestern Kansas has just truly been devastated by drought. The breeding programs that serve the state of Texas lost every single trial site that was not irrigated in 2022. They were completely unharvestable. So drought is having very real impacts on production in our region, despite these very high precipitation events. So what do I see from the vantage point that I have in Manhattan, Kansas is the effects that we really need to work to address now. The first one is changes in stripe rust, as we call it in the United States, or yellow rust. The second is the resistance to the Hessian fly, the barley midge, and the third, of course, are the heat and drought effects. And then last, I'll talk about what some of the impacts are for wheat production in the context of greenhouse gas production constraints and high energy prices. So in terms of breeding for stripe rust or yellow rust, and I want to thank Dr. Garcia-Leon for her presentation because she really set the stage for this discussion. Before 2010, the economic impact of stripe rust was really negligible on the Great Plains. It was much less important than leaf rust. Economically important epidemics were rare. And that was because stripe rust really was inherently limited at warmer temperatures. Stripe rust was a cool weather disease. And so the breeding programs throughout the region focused on slow rusting genotypes, quantitative adult plant gene combinations that could really get the crop through until the weather warmed up enough to shut the stripe rust down. But after 2015, stripe rust became the most economically significant rust disease, causing upwards of 15% yield loss in some years in Kansas. And that's because the pathogen has, in fact, evolved to tolerate higher temperatures. And this is work that's been really nicely demonstrated by Eric DeWolf, a pathologist at Kansas State University, that the pathogen is, in fact, not shutting down at the temperatures that previous pathotypes would have. As a result, the slow rusting adult plant gene combinations that we've come to rely on really now are insufficient. So we've made a hard pivot in our breeding strategy. On the left side is a plant that actually has a slow rusting gene combination, and it's clearly severely impacted by the presence of disease in the field. And this impact was a plant under natural infection conditions. On the right side, you'll see here on the left are susceptible spreader rows. And then this block of materials is a block of breeding germplasm that has the combination of the major all-stage resistance genes YR5 and YR15. This combination of genes has not been deployed in the Great Plains to this point. Both of these genes are still highly effective in the Great Plains, and we see this durable combination of major gene resistance as really a path forward for stripe rust in the region. The YR5 and YR15 combination isn't the only one in our breeding pipelines, but it is one that we've really dedicated significant effort to bring online, because we see this as a way to help us address the change in the pathogen dynamics in its interaction with the wheat crop in the context of climate change. The second biotic stress factor I'd like to talk about is the Hessian fly. The Hessian fly is an important pest in plains. It has tended to be episodic. This last year in Texas, several counties lost vast areas of their wheat production to the Hessian fly. The female fly lays her eggs and they hatch, and the larvae move down between the leaf sheath to the base of the plant and feed on the growing seedling. You can see in the right here some of the larvae and how they feed, and they really can cause tremendous stand losses. There have been a number of resistance genes that have been developed for Hessian fly. The fly tends to rapidly overcome these in a gene for gene manner, which is problematic. Producers have tended to rely on avoidance strategies for this pest. There used to be what we knew as a fly-free date, dates after which the flies were no longer active. With the warmer fall temperatures, those have really ceased to be effective determinants of a safe planting interval. The other issue in our growing region is that we have seen an increase in, and I apologize for the typo, it's what we call graze and grain production. Producers, particularly in Oklahoma, will plant winter wheat early to produce forage for cattle, and then remove the cattle in late winter, early spring, and produce a grain crop. And this graze and grain production system has led to increased plantings at earlier and earlier dates, and that's really promoted the exposure to the Hessian fly. Most of the resistance genes that we have are ineffective at temperatures above 25 centigrade. So we've needed to look to find high temperature resistance genes that are effective above 25 centigrade to provide options for producers for planting in the fall. And this work is really a result of effective international cooperation. Back about 2008, 2010, Dr. Ming Chen and our unit cooperated with scientists at ACARTA and brought in Durham land races to screen those land races for high temperature resistance to Hessian fly. And in fact, identified a set of 5 Durham accessions that had resistance to Hessian fly at 30C. And the work that I've done has been to pick up these land races and then introgress that resistance into adapted hard winter wheat germplasm. So in this table, we're looking at the number of seedlings that are resistant relative to the number of seedlings that are susceptible to the Hessian fly. And Zenda, our recurrent parent that's susceptible, is predominantly susceptible in the greenhouse at 20 centigrade and in the growth chambers at 25C and at 30C. Molly, which has the H13 resistance, which is a very important resistance gene in the region, again, is highly resistant at 20C, at 25C, but at 30C, its resistance completely collapses and it's fully susceptible. One of the introgression lines that we've developed by backcrossing Zenda to one of these ACARTA land races demonstrates resistance, though, at 20C, 25C, and 30C. And this is really a very promising result for this resistance. We've also seen resistance from other Durham land races that's providing partial resistance at these warmer temperatures. So we're thinking that we may actually have 2 different resistances here, both of which might be able to be complementary and really help provide that high temperature resistance to this important pest. Our unit also has a long-term effort in breeding for heat stress. And there have been really two approaches to this kind of work. The first is work done by Dr. Zhanming Fu, who's been connected to the unit for many years, who has done greenhouse and growth chamber studies looking at QTLs for chlorophyll retention under heat stress, essentially stay green traits in wheat. And he's identified in a number of association mapping populations and in biparental populations, some very strong QTLs for chlorophyll retention under heat stress. We've also, in my program, taken one of these populations out to the field and looked at grain weight retention under heat stress, because I really see that ability to fill the kernel under heat stress is really a primary indicator of high temperature tolerance. And again, we're able to find significant QTLs within adapted germplasm for that ability to retain grain weight retention in these stress environments. The new initiative, though, that I really want to transition to here is to utilize the genetic variation in wheat's ancestor, Trichome dicocoides, which we know as wild emmer, as a source of tolerance to both heat and drought stress. This is a bit of a replay of Dr. Cloutier's presentation here on the evolutionary history of wheat, which is, of course, a polyploid derived from a series of hybridization events and then some domestication events. The wild emmer is here, has both the A and the B genome. It is the ancestor of modern day durum wheat. Okay. And it was the hybridization of derivatives of the wild emmer with the goat grass ancestor that led to hexaploid wheat, followed by subsequent domestication. So really the ancestor that we're focusing on here is Trichome dicocoides, the wild emmer. And what we know is that, and this is from work in the Akonoff lab at Kansas State, that this path of domestication is not entirely a one-way top to bottom, that in fact there has been some level of crosstalk back of the wheat germplasm to the wild emmer, but a relatively small proportion of the wheat genome at this point really can be assigned to wild emmer. So although there's been some crosstalk, it hasn't necessarily had as much impact as it might have if we went back and intentionally did some plant breeding here. So the work that we've been doing, and I've got this slide on the background that is used for the NSF Center at Kansas State University. It's an industry university cooperative research center that incorporates the Wheat Genetics Resource Center at Kansas State, Colorado State, and a number of private industry partners. And so as part of this partnership of the WGRC with our agency and Kansas State and Colorado State, we have accessed a mini core collection of wild emmer from the WGRC. So this is a genomically curated collection. And we have crossed those wild emmer accessions to the hexaploid breadwheat, produced the pentaploid F1s, which are largely sterile, and then back crossed those F1s to the breadwheat to produce back cross hybrids. And we have then advanced those hybrids by single seed descent to develop what is effectively a mini nested association mapping panels through the F5 generation. So we're trying to create here introgression libraries that capture the diversity in the wild emmer ancestors in adapted wheat backgrounds. And the thinking behind targeting the wild emmer is that the wild emmer collections really have come from some of the hottest, driest, most challenging environments in the world. They're collected from Israel, Jordan, Syria, Turkey. So we're really hoping in this germplasm to recover alleles for heat and drought stress. It's been a labor of love to work with this because wild emmer itself has a brittle rachis. It shatters at physiological maturity, and it has tenacious glooms. So it's kind of a wicked combination of plants that shatter and don't thresh. Nonetheless, we have been able to successfully advance wheat germplasm to the point that we're now looking at this germplasm in whole plot field trials, and we're able to begin to feed this germplasm in batches through some of our cooperative testing programs. An example of this is stripe rust evaluations by Dr. Sean-Ming Chen in Pullman, Washington. Another example is pieces of this have been included in the Kenya stem rust screening nursery. And we're now at the point of launching physiological studies of these under really intentional stress. So it's really exciting to start to see this germplasm come forward to the point that we can evaluate it. We're coupling these evaluations with some high throughput phenotyping by UAV, which is also really very, very interesting for us. One of the other features or facets where this germplasm is helpful to us, I think, is in addressing the breeding for some of our production constraints, specifically the requirement for synthetic nitrogen fertilizer in wheat production. Protein very much matters in hard wheat. This graph illustrates the bread loaf volume in response to flour protein. This is about 8,000 bakes of the regional nursery. And we can explain 40% of the variation in bread loaf volume on protein alone. The problem is that as yield increases, as we see in the graph on the right, grain protein concentration decreases. So wheat breeders in this market class really have been fighting the demand for higher protein while trying to maintain high yield levels. And, you know, it's really one of the great challenges of breeding hard wheat. This is important to producers because if we look at the proportion of operating costs for our producers that are attributed to fertilizer applications, for wheat producers, about 35% of their operating cost is actually synthetic nitrogen fertilizer. So this is a really important fertilizer use efficiency is really important to our producers. And fundamentally, fertilizer is where the largest share of the climate impact of our wheat production comes. So developing wheats that better utilize nitrogen fertilizer are important both to mitigate climate change and to respond to it. So within the wild emmer introgression germplasm, we've started looking at essentially the quadrants, those wheats that yield above the checks and those wheats that have protein higher than the checks. And this right-hand quadrant here really is the target that we're breeding for with recurrent selection in this germplasm, to breed for that combination of high yield and high protein. Finally, you know, as much as we breed for climate change, we're also breeding in the context of climate change. And that's meant changing how we do what we do. And I have a little bit of reflection on that, you know, in kind of a real-time look at this. Collaborations have become increasingly important. I'm leaning into the Colorado State University physiology team that's actually doing work at a USDA facility in Greeley, Colorado for drought stress characterization. I'm looking for collaborators on heat stress evaluation on this germplasm, that I'm less and less able to do myself all of the things that need to be done to capitalize on the variation that we've been creating. Secondly, risk management has become a tremendously important component of the program that I operate. we have phenomenal trial site losses. In 2022, we planted 9 locations and harvested 5. We had an extreme rainfall event, we had frost, we had brown mites, we had hail, we had herbicide drift. So my interest in a breeder in very large, high stakes trials has become very limited, that I'm looking to diversify the risk in my testing program. And finally, to evaluate breeding germplasm, you actually have to be able to produce seed. And we're no longer at a point where we can reliably produce seed for trials without irrigation. So that has meant leaning into facilities in our agency that have irrigation capacity, whose focus is not breeding and genetics. The Meat Animal Research Center north of us in Nebraska has irrigated acreage, and graciously they've agreed to cooperate with us towards seed production. The Limited Irrigation Research Facility in Colorado, again, similarly focus not breeding and genetics, but an agreement to cooperate and to work with us toward helping us really be able to produce seed and be able to produce trials to address these challenges. I'm definitely not alone in my rate of loss of trial sites. This is impacting every breeding program in the region. So with those reflections, I do want to acknowledge the tremendous collaboration that I have. Alan Fritz, who is the wheat breeder at Kansas State University, and Edward Akinoff, who now directs the Wheat Genetics Resource Center, have just been fabulous cooperators to work with, as has the whole WGRC team. And it really takes a village to do the work that we do. And thank you.
Thank you so much, Mary. And you know, you bring, unfortunately, we don't have time for questions, but please, if you do have questions for any of our speakers, put them in the chat and they can answer them directly in the chat or reply with an email to you. So please continue to think about your questions. And Mary, you brought up a great point, collaborations. And I want us to think about not just collaborations within the US, within Mexico, within Canada, but across countries. And that's what Prosa Norte is all about, is bringing people together for those collaborations. And so thank you for mentioning that, because it is a great point and a necessary point nowadays for the research that we all are trying to do to mitigate the effects of climate change. So thank you very much for your presentation. Okay, so we are on our final speaker presentation today, and it's my pleasure to introduce Dr. Camille Stieber, who obtained her PhD in molecular genetics from the University of Chicago. She did her postdoctoral research at the University of Toronto, studying the hormonal control of seed dormancy and germination. And she is currently a USDA ARS scientist with the Wheat Health Genetics and Quality Unit in Pullman, Washington, in the United States. And she will be speaking today about the effects of rising temperatures on falling numbers and pre-harvest sprouting. So Camille.
Thank you for the kind introduction. So I'm a USDA scientist located in Pullman, Washington in the United States, where I collaborate with scientists at Washington State University to identify genes and breed for tolerance to low falling numbers in pre-harvest sprouting. In the process of doing this research, I have encountered problems or made observations as to the effect of increasing temperatures on tolerance to pre-harvest sprouting and low falling numbers that I will tell you about today. And for some strange reason, my slide's not advancing. There it goes. Okay, so Washington State is part of the US Pacific Northwest. where most of the US soft white wheat is grown, shown in blue dots here compared to the rest of the nation. Soft wheat grain has low protein and good baking quality for cakes and cookies. This is also the only area of the US that grows club wheat, a type of soft white wheat known for its exceptional cake quality. So we are interested in falling numbers because it's the method that the wheat industry uses to detect alpha amylase production in wheat grain. Higher levels of alpha amylase result in higher levels of alpha amylase detected based on low falling numbers, results in sticky bread, which can gum up the knives when it's sliced in a bakery, causing financial losses for bakers. Increasing alpha amylase can also result in cakes that fall. Most of our soft white wheat is exported to Asia, roughly 85% of it, where it is quite commonly used to bake gift cakes, these sponge cakes, such as this puffer fish shown here. If this cake had fallen, the puffer fish would look very different. So as you can see, our customers care a great deal about quality and about having high falling number wheat. So you can't just knock out wheat alpha amylase. It's a very important part of plant physiology and is expressed as a normal part of wheat grain development and germination. Alpha amylase levels are high as the wheat embryo develops, and then once development is finished and we go into the grain maturation phase of development, those alpha amylase levels drop as grain filling occurs and the wheat endosperm is formed. Also during grain maturation, the grain acquires dormancy, which prevents germination under favorable conditions and desiccation tolerance so that the grain can dry down, which is good for storage. So if you have high dormancy, if you have low dormancy and we wind up with a rainfall event before the wheat is harvested out of the field, you wind up with this condition called pre-harvest sprouting, the initiation of germination on the mother plant when rain occurs before harvest. So alpha amylase is expressed during germination and during pre-harvest sprouting because it mobilizes the starchy endosperm as a source of food for the growing seedling. Alpha amylase can also be produced during development in response to cold stress during the soft dough to hard dough stage of grain maturation. This phenomenon is called late maturity alpha amylase. I will focus first on the effects of climate change on pre-harvest sprouting and then say a few words about LMA at the end of the talk. So seed development and germination are exquisitely regulated, right? The decision to germinate is really important to the success of a species. So it should come as no surprise, the temperature affects these processes at multiple stages of development. Cooler temperatures during grain maturation result in more dormancy. Nakamura et al. Have shown that this is associated with higher expression of a dormancy gene called MFT in wheat, mother of flowering time in TFL. This gene was also mapped to chromosome 3A as an important QTL for pre-harvest sprouting tolerance, both by Nakamura in Japanese germplasm and by Guiwai Bai in Kansas germplasm. Dormancy, we should wind up with lower dormancy if we have higher temperatures during grain maturation. This will lead to increasing susceptibility to pre-harvest sprouting if we have premature grain before harvest. Interestingly, work from Lay et al., where wheat was planted midsummer in order to make it first graze wheat as grass before it goes through vernalization in the winter. But they found that when wheat was planted in the summer, sometimes those high temperatures resulted in a form of thermodormancy that caused failure to germinate. When they mapped this QTLs for this thermodormancy, they also landed on the MFT gene. So it looks like MFT is both associated with dormancy triggered by cool temperatures during development and by dormancy associated with high temperatures in mature seed. If we wind up with a warmer climate, we may find that in order to plant wheat at the cooler temperatures needed for efficient germination, winter wheat may need to be planted later in the fall and spring wheat earlier in the spring to avoid problems with poor emergence. Dormancy is lost through the process of after ripening. Dormancy is highest at physiological maturity, just as grain filling ends and the wheat turns yellow. As the wheat is subject to dry storage or stands on the mother plant, PHS tolerant lines lose dormancy more slowly than susceptible lines and acquire germination potential and sprouting susceptibility more slowly. Higher temperatures also speed the after ripening process. So it may mean that we will have less time to get the wheat harvested out of the field once it reaches maturity. So as I said, pre-harvest sprouting is the germination of grain on the mother plant when rain occurs before harvest. Resistance to pre-harvest sprouting is largely a result of seed dormancy. It accounts for 60 to 85% of tolerance. However, spike morphology can also contribute to pre-harvest sprouting tolerance, especially traits that keep water out away from the seed, such as tighter blooms, more surface wax, or lack of awns. While dormancy is good for preventing pre-harvest sprouting tolerance, as I mentioned, if you have too much dormancy, it will give you problems with poor seedling emergence. Poor seedling emergence will have a negative impact on yield. So breeders have to balance between having enough pre-harvest sprouting tolerance, enough dormancy to prevent sprout, but not so much that they wind up with problems with poor emergence. So pre-harvest sprouting can cause financial losses based on how extreme the case of pre-harvest sprouting is. I mean, if you have a really extreme case where you have seedlings growing out of the spike like this, you're going to have almost total yield loss, right? It might not be possible to harvest the crop if the roots grow back into the soil. I had a farmer in Washington tell me that that happened to him back in the 80s. In a more mild case where you see visible sprouting, you might be able to sell this wheat as animal feed, which would also give you loss of yield. But interestingly, it's possible to have very early stages of germination prior to the point where you actually see visible emergence of a seedling, where you have elevated alpha amylase. So here is an example of a sprouted grain of the cultivar Briel, where you just see some expansion at the embryo end compared to the sound sample. This wheat had elevated alpha amylase, resulted in starch degradation and a low falling number. So the falling numbers test was really invented as a way to catch these sneaky guys, right, that have just that have elevated alpha amylase without obvious germination. The falling numbers test is based on looking at the loss of viscosity as you get starch degradation by alpha amylase. Large starch chains have good gelling capacity and make a viscous gravy. Once alpha amylase cleaves this to shorter starch chains, you get less and less gelling capacity and a more watery gravy. During the falling numbers method, we make a wheat meal water slurry, and then is then heated and mixed to form a gravy and a watery gravy. Then falling numbers test measures how much time it takes in seconds for a stir to fall through that gravy. A watery gravy with a lot of alpha amylase and starch digestion allows that plunger to fall quickly and gives you a low falling number. A more viscous gravy that has low alpha amylase will give you a higher falling number because the plunger falls more slowly. So in Washington State, farmers receive a discount based on falling number of 25 cents for every 25 seconds below 300 seconds. That may sound like kind of a forgiving way to do it because it gives an incremental discount, but it can have a devastating effect when added up. In 2016, our falling numbers problem cost Washington growers on the order of $140 million. You can see that we are having more frequent problems with low falling numbers in the Pacific Northwest after roughly a 10-year hiatus with very few problems with low falling numbers. I can't say for certain that this is due to climate change because there were a lot of problems with low falling numbers during the 1980s. What I can say is that the falling numbers test can protect millers and bakers from financial loss, but the only way we can protect farmers is by breeding for genetic resistance and identifying resistance genes. We've been doing this in collaboration with wheat breeders in Washington State, making use of two assays for pre-harvest sprouting tolerance. The spike wetting test, in which we harvest grain from the field at physiological maturity after ripened for 5 days, so they all have the same degree of dormancy loss, then place the wheat spikes under a misting system, mist for 6 seconds per minute, and then score daily for 7 days based on this visual sprouting scale developed by McMaster and Diarra. Scores of 1 through 5 basically show you increasing degrees of germination in the strict sense, whereas scores 6 to 10 are looking at post-germinative seedling growth, the speed of that growth. This test measures the combined effect of grain dormancy and spike morphology. When we want to look at dormancy alone, we make use of germination assays on plates, again with 5 days of after ripening after physiological maturity, and we score based on percent germination. So our initial efforts to identify pre-harvest sprouting tolerance genes were in 2 genome-wide association studies in related panels of Pacific Northwest wheat. I was a little surprised to find that only a single QTL overlapped between the 2 studies. This QTL was on chromosome 2D and co-localized with the compactum or C locus that determines club head type. Since the C locus is very close to the centromere, there is a low level of recombination. So in our population, we found that the tolerant allele of 2D was strongly associated with the lower sprouting scores in our populations. So we didn't see the recombination between the 2 loci, so it's difficult at this point to know whether our QTL is the C locus itself or a neighboring locus. The other question that we had was whether pre-harvest sprouting tolerance from this QTL was due to increased seed dormancy or due to the compact spike morphology of the club wheat. We asked this question, or rather Jason Wiggin asked this question using a biparental doubled haploid population derived from the more dormant club wheat Cara and the lax wheat Zerfa. He reasoned that if pre-harvest sprouting tolerance came from club morphology, we would detect it only by spike wetting test, not by germination assay. If pre-harvest sprouting tolerance came from dormancy, then we should detect the phenomenon with both assays. That's actually, in fact, what happened. He found that the 2D QTL was a very strong QTL with a maximum LOD score of 12, explaining 30% of the variation. What was curious about our study was that none of these studies mapped the MFT gene on chromosome 3A. In fact, based on CASP assays developed by Bai, we were able to find that our population was monomorphic for the pre-harvest sprouting susceptible version of MFT at minus 222 in the gene. So what this tells us is that it is possible to identify QTLs that seem to be able to function in the absence of MFT. Because temperatures were increasing over the 3 years of Jason's study, we were able to look at the effect of increasing temperatures during grain maturation in his population. So this shows you the timing of grain maturation with the blue box. It was warmer during maturation during 2020 and very much hotter during 2021. It's a little difficult to see on the stick graph, so I showed you here number of days over 37 degrees Celsius, number of days over 32 degrees Celsius in the 3 consecutive years. Unfortunately, we didn't harvest our population in 2021 because we had a lot of trouble with pinching in one sector of the field. But you can see that our population, comparing 2019 to 2020, looking at sprouting score with increasing days of misting, we had similar distributions of sprouting score at days 3 and 4 of misting, but by 5, 6, and 7, we were seeing much higher degrees of sprouting in the warmer year 2020. Jason was able to look at sprouting scores in our highly pre-harvest sprouting tolerant check Clark's cream. Clark's cream showed essentially no difference in sprouting scores in 2019 and 2020, suggesting that it does have a fair degree of resistance to heat during maturation, but it did show increasing sprouting scores on days 3, 4, and 5 of misting with the extreme heat in 2021. So the fact that this line has some degrees of tolerance, I think suggests that we should be able to find sources of pre-harvest sprouting tolerance that will be more heat resistant. This is especially going to be important if our higher temperatures are associated with more rainfall, as was mentioned about predictions in Canada. The higher temperatures that we saw in 2021 also allowed me to ask some questions about the effect of high temperatures on late maturity alpha amylase. Cold induces LMA, or the expression of alpha amylase, when it's experienced during the soft to hard dough stage of maturation. A study from the UK and another study from Australia suggested that high temperature shock during these stages of development can also cause LMA. So we were a bit worried about this when we saw our high temperatures in 2021. We were able to ask this question using spring wheat that we planted that had 3 different types of characterized LMA phenotype. Resistant, where cool temperatures did not result in increased alpha amylase activity. Cold induced, where cooler temperatures during grain filling did induce alpha amylase, and LMA constitutive lines, where LMA is essentially always on, or at least so we thought, on at our higher temperature of 25 degrees day, 18 degree night, and at our lower temperature of 18 degree day and 8 degree night. This wheat of these 3 different phenotypic categories was planted earlier on April 1st and then later on May 3rd, allowing us to look at the difference between a line that experienced heat earlier during development versus a line that's experienced the heat later in development. And here we're looking at falling number, where a low falling number means more alpha amylase. So when I first looked at this data, my first thought was, wow, they're right. More heat gives you a lower falling number. Maybe this is LMA. And then I started to realize that, wow, look at this. Our LMA constitutive lines are showing falling numbers above 300 seconds when they were planted on April 1st. If you compare that to how our lines behaved in 2020, you can see that the heat experienced in 2021 with the early planting consistently resulted in higher falling numbers indicative of lower alpha amylase, and that indeed it rescued our LMA constitutive lines to a falling number well above 300 seconds. But that later planted wheat had very high alpha amylase and low falling number. What was going on? When Galina took a look at this wheat that she'd run falling numbers on, she discovered that the heat resulted in shriveled grains, some of which were green in color, which caused us to speculate that heat may have been causing the wheat to dry down and die before the completion of grain filling and maturation. This may have prevented the alpha amylase that's expressed early in development from decreasing, resulting in elevated alpha amylase, not because of LMA, but because we had developmentally challenged, pinched, shriveled grains. I suppose if we get this kind of heat, falling numbers may be the least of our worries. We're really going to be more worried then about yield and test weight. So our conclusions: heat may decrease pre-harvest sprouting tolerance, but improve LMA tolerance. But we have to be careful of the caveat that these processes are impacted by multiple environmental factors at multiple stages of development. Our new USDA scientist, Jean-Ron Lee, plans to look at this by modeling the impact of multiple environmental effects, environmental factors, including temperature and moisture on pre-harvest sprouting, LMA, and falling number. We were able to make use of club wheat to identify a unique source of pre-harvest sprouting tolerance. This tells us that it could be very useful to screen diverse germplasm for QTL that provide heat-resistant pre-harvest sprouting tolerance. I know the USDA scientist Brian Penning in Worcester, Ohio, is screening land races. It could be a good idea to collaborate with Sylvie, who talked earlier about looking at improvement through synthetic wheat. Our data show us that it is possible to identify QTL that seem to be able to function in the absence of MFT, or in the absence of a beneficial allele of MFT. We need to take a closer look at MFT, I think, to see if it really is a master regulator controlling the effects of heat on dormancy and pre-harvest sprouting tolerance. If it really is, then MFT may be a good target for pre-breeding, either through genome editing or through identifying new natural alleles. I want to acknowledge the people in my lab who did the work and our collaborators at Washington State University, Aaron Carter, Jiwoo Zhang, Michael Pumphrey, Amber Hovermill, Scott Neely, who runs the variety trials. Kimberly Garland Campbell is responsible for pointing me in the direction of club wheat, and the WSU and USDA field crews. I want to acknowledge funding from USDA RS, from the Washington Grain Commission, and from USDA NIFA. With that, if I have time, I'll take questions.
Thank you very much, Camille. Unfortunately, we don't have time for questions, but again, I encourage anyone, if you do have questions, to please put them still in the chat. We have just some closing remarks, and or reach out to contact us.
I will hang out for a while afterwards. Maybe Mary will too, just in case people want to have a discussion after the session ends.
Thank you. Yeah, if you can stop sharing your screen, and then so again, I want to thank all of our presenters. All did a fantastic job of really highlighting the challenges that wheat growers are facing with climate change, and the techniques and strategies that are being used to combat it. So, and I want to also just acknowledge and give my appreciation to our two translators, Cynthia Diaz and Luciana Pavez, who have done a fantastic job of keeping all of us able to understand the presentations in the different languages that are given. So thank you very much for that. And so now it's my great honor to introduce the Executive Secretariat for IECA, Dr. Jean-Charles Lavalier. And Jean-Charles is going to give some closing remarks, and he will have the final word. And I will say one more thing just before. I encourage, again, Thursday, we will be having similar sessions, different speakers on grapes. So please plan to attend that as well. And Jean-Charles, I turn it over to you now. Thank you.
Thanks, Tim. Well, instead of talking about pufferfish case, we're going to be talking about wine on Thursday, probably. We'll see. But I wanted to join you on my thanks. I just so that everyone know, I'm the Executive Secretary for Poste Norte. And so as part of ICA, I'm based in Ottawa. I'm the country representative for ICA here in Canada. And so my sincere thanks. I joined Tim and the task force on congratulating them on putting a fantastic agenda, great speakers. Thanks to all the speakers. Thanks to everyone who are taking part out of your busy schedules to be here today with us. And you know, I've learned a lot. It's always enjoyable. Brings me back to my plant health days when I was studying as an agronomist. And so plant health science is really important. Your work is so important in wheat. And generally, in terms of plant health science, as more broadly, it's a discipline that's very fascinating in terms of the uses. And like what Tim was saying about helping our growers, it could be anything also from measures to control and prevent pests. It could be anything to do with human interactions. And yeah, as you know, pests can be invasive. It's not just pests, but some of them are well established, well known. Some are emerging. There's some new variants. And so I'm thinking for us at ICA, plant health plays more role broadly in terms of sustainable agriculture, in terms of food security, in terms of our practices as our growers, in terms of agriculture practices, and even sustainability principles. And plant health plays a critical role in anything to do with climate change and the topic of today. And I feel that overall, in terms of the subject matter that we saw today, I felt part of your village, as one of your speakers said. I've been enjoying the research. I've been looking at trying to understand the data, understanding some of the results, the projections, and I had not thought of. I actually had never heard of the bakeries in Asia and how wheat was used. I bake cakes all the time, but that was new to me. But the interactions on yield, on productivity, on the protein levels, that's so important. And the importance of wheat into the world economy as a major crop, one of the 5 really top most important crops on the planet. So job well done. My thanks to interpreters, my thanks to the logistics team here in Canada, to the task force, to Dr. Kapis, our president, for his opening remarks. And really, I've been very thankful to the whole of the speakers. You put a lot of effort and invested time in your presentations. It shows. And my thanks again to all of you. Please stay safe. A reminder again that this webinar is being recorded. We tend to usually put these we post it on our ICA Canada webpage in Costa Rica and on our Poste Norte website. So it lives on. So please stay safe. A reminder again to come back on Thursday, and we'll have this same place, same time, online, and it's about grapes. So please have a great day. Enjoy your evening. Thanks again. Cheerio, everyone.